So first of all, I would like to welcome all of you about today's course, which is generally about meta-analysis and some new techniques. However, I think it's really very important before we start speaking about one method in science to understand what is really science and what is translational science, translational medicine, because I think at that moment this is mixing almost in every people what is science, whether meta-analysis is a science, whether and how to move from the bedside to the bench and back to the bedside. So first of all, question number one, I think it's very important, what is science? What we actually call for science. If we look at the different books and the different articles, we must say first that this is a systematic enterprise that builds and organizes knowledge in the form of testable explanations and predictions about the universe. So this is very importantly a systematic enterprise, and always doesn't matter where you are in science, you always need to perform as a good scientific practice, which very importantly must be objective, and the other must be reproducible. This is very important, and if a paper is published in any kind of scientific part, these are the major points what you always need to put in front of you. So science, to find out something which is unknown, you need to take it to the known part, and what is the enterprise? How can you go actually from the unknown issue to the known one? First, you need a question. Plenty of times we can see in science that somebody is doing something and the main aim, because it hasn't published yet or nobody looked at yet. Very importantly, you need a distinguished question, and you always need to ask from yourself, yes, this is the best question I can raise in this topic. This is really important in the field. Second, you also need to have a hypothesis. It doesn't matter that finally you will be right or you will be wrong. Actually, the two most important discoveries we have published, the hypotheses were wrong at both cases, and finally directly the opposite results came out. But first, you need to idea which way you would like to go forward. The third issue, and today meta-analysis is one of the methods you can use in science, about you need a methodology. You need to follow a very structured methods to get answer to your questions. And if you have the methods, you need to collect data. There are several ways how you can have data. You can do basic experiments. This will produce data. You can do clinical trials. You can have data from the trials. Or, for example, in a meta-analysis, you collect data from the literature. But data collection is really very important in science. And, of course, when you have the data, you need to analyze the data. So biostatistic analyses are really crucially important. And finally, if you start from here and you go further, you can get answers. Also very important that this is not a one-way enterprise, because sometimes when you collect the data, you realize that something you didn't do well, and you turn back and you change the methodology, or you change the hypothesis, or when you had an analysis, you realize that your question is much more difficult to answer, and you're going around it, but you have to follow these kind of structures. So the next question, this is about science. Doesn't matter it's a social, medical, or any kind of science. The second question is, how can you answer, or how can I answer questions in medicine? From questions to answer. There are three major scientific ways how you can answer. First, basic science. We call basic science, which is pure fundamental research. You try to find an unknown mechanism. Second, when you have this mechanism and you know it, the second part is the applied science, which develop knowledge for practical application, so developing the knowledge from basic science. And the third one, when you have a practical application process, you try to use it in clinical science. What are the aims of the different procedures? First, the basic science, discovering new mechanisms and targets. This is one of the major aims. The applied science is developing new drugs and intervention based on these kind of targets. And the third one, when you have your drugs or different interventional part, to understand the safety and effectiveness what you have. So if you have a questions, you immediately need to understand which part you need to join in. For example, if there is already, you have a drug, but nobody used in the disease, you would like to test. And you have the questions, for example, whether biological therapy is good in pancreatitis, what is already shown that good in IBD, but nobody tested in pancreatitis, you join here. But if you would like to understand the question which never ever studied, and you first need to understand the mechanism, how the disease is developing, you go here. So this is depend on where you need to go in, not whether you are a basic scientist or a clinical scientist. This is always a problem, that you have a question and you try to go to the way in which you are and not thinking about which part you need to enter to the question. And the third one, which is really important, there are huge amount of methodologies, and I just put here some of them. For example, in basic science, in applied science, and also understanding clinical science. So you have the different sciences, you have major aims, and you have methodology. And of course, this is again not a one-point enterprise. You always go one to the others. Look at how can you use this scientific information. You get the answer to your questions. First, if you are in a theoretical department, you are in anatomy, physiology, molecular biology, chemistry, you teach it for medical students or postgraduate course. So the science you have, one way you can use it for educational purposes. What about the other part? Of course, if from science you get an evidence what is better for the patients, immediately you can put it to clinical practice. This is called evidence-based medicine, using medicine only, which is scientifically proved that the patient has benefit from it. And of course, you can use it also for teaching activity and also in the patient's care. Let's look at what is only most probably for basic scientists. Of course, if you are in a department, anatomy, physiology, or the others, and you are only in the basic parts and you know only the basic methodology, usually like using cell imaging in microscope, working with animal models, or doing molecular biology, you most probably using this part. If you are a clinician, of course, you can perform different randomized clinical trials or observational clinical trials, but of course, who is working in the department of anatomy, for example, of course, they cannot do a randomized clinical trial because they don't see patients. But who is here also don't go under the microscope. However, there are other periods which is very important and could be really very useful for both sides. First of all, understanding about the pharmacology or the different pharmacokinetics, dynamics. This is so important for clinicians and also important for basic scientists. And also in a clinical science, there are analyzing methods which you don't need to involve patients, you just need the data. For example, meta-analysis, or if you have a prospective database, doing data analysis, or you can do, of course, retrospective data analysis. So today, meta-analysis will be one of the methods in science. What both sides, doesn't matter you are in basic or in clinical science, you can use. Okay, so you could see about how complex is how you can answer the question. So the next question is, what is translational medicine in this big enterprise of science? So translational medicine is there are various existing definitions. So if you go and try to look at what you mean translational medicine, plenty of different data you can see. However, if I think what is actually the really best definition for it, this is first of all very interdisciplinary, and the main aim to speed up the process of moving any kind of research discoveries for patients' benefit and using the data with absolutely no restrictions. So what does it mean? That translational medicine is not only from bench to the bedside, because most of the people thinking that. Also, if you find any kind of scientific data, even from meta-analysis, different cohort analysis, and you immediately speed the process up and put it to clinical use, this is also translational science, because translating scientific results back to the patients, and the main aim of translational medicine to speed the whole process up. So first, why do we need translational medicine? I usually say when I speak about translational medicine that this is back to the future. Because if we look at before the 50s and 60s, before the big developments of different physiological techniques and different clinical techniques, medicine were together. So the same people who was working in the clinic did the experiments in the laboratory and brought the results back. If you think about, for example, Semmelweis, who was also in a gynecology department and the pathology department and were together. However, after the 70s, when plenty of basic techniques came, imaging, genetics, proteomics, everything, and also in the clinical, huge amount of different clinical points came. The two parts came absolutely separately, and now you either go to a basic science department and never see a patient again, or you be a clinician and never do any kind of basic research any longer. So the two periods went away from each other, and if you look at what is actually translational medicine, is to make actually a connection between different interdisciplinary parts, not only in medicine, but also in mathematics, also in IT, and everything. You put together the different sub-disciplinary way. So who wins by translational medicine? This is always an issue, that whether shall I join or put to my patients to a translational way. It's very easy. For me, in science, this is an absolutely win-win-win situation, because everyone who is in the system will win. Let's look at the patients. For example, here is one of the results that we realized with basic scientific knowledge. Also, we realized with meta-analysis, cohort analysis, that early energy replacement for patients suffering from acute pancreatitis needs early energy intervention. So because this is already were in the clinical use, we change the process, and we don't start the enteral feeding only when the patient has severe pancreatitis, but we start immediately after admission. This only change meant that we could reduce the severe and moderate pancreatitis. In average, the average days of hospitalization decreased with 2 days. So for example, if we see 200 patients a year, this means less than 415 days hospitalization, for example, in one disease. Think about how many diseases we treat in a clinic. And if you could achieve that all patients stay 2 days earlier, how much benefit this is for the patients and also for the economy. Or if you look at also like the mortality level, the usage of antibiotics, the necrosis, all clinical data clearly shows that, for example, from basic science to meta-analysis, everything, bringing the knowledge immediately to patients can have to patients benefit. Let's look at the healthcare budget. I think you can realize that if the patients are in the hospital for less days, you use less amount of medicine. This is also really changed the budget. For example, if you look at the general center with the translational pancreatic centers, and you look at how much euros you spend on one patient in the different diseases, and you count an average cost of pancreatitis per patient, you immediately can see that if a patient in a translational part get less drugs, the patients, the disease outcome is better, and the patients are less amount time in the hospital. The whole economy is cheaper. And at that time, if you show these data to the different insurance people and company, you may change the finance issues for a disease. What about the individual scientist? In January 2016, in Page, we established the Center for Translational Medicine at the very first time in Eastern and Central Europe. We started working on translational medicine on 2 departments. One of them was the Department of Pathophysiology, which was earlier only in a theoretical department, and the other was the Department of Gastroenterology, where they saw patients, but they didn't went back to understanding the basic things. I just would like to highlight about the CVs of the different people, and here is about the impact factor, how it came to their actually CV, and also you can see about the citations, which is, of course, lower starting, because this will followed by the impact factors. If you look at the different people in the Department of Pathophysiology, you can see that all people who make the connection between basic and clinical science and use the different methodology all together. So ask the questions and choose the methodology and went back, and not first choosing the methodology, try to find the question what I can answer on my methodology, because this restricts the results what you can. And also if you look at the PhD students, like within 1 year, they also can achieve many publications about that way. So if I look at the different CVs, scientists also can win. And if you look at the institutions, in an institute there are, I must say, 2 leaders. One of them is the counselor, who is thinking about money, and about the rector's office, who is thinking about achievement and impact factor. Of course, you need to satisfy both parts, because you just cannot go there to say that how many impact factors you have. In translational medicine, we could won 1 GINOP and FOP grants, and both of them more than 1 billion Hungarian forints came to the University of Pécs. And if you look at the different going up of the different departments within 2 years, you can see that you can satisfy with translational medicine also the University of Leader. So the next question is about, if I look at the methodologies, if I look at Eastern and Central Europe or Hungary, what are the methodologies are in a curriculum in the medical education, what we already know. If I look at these parts are usually covered, and if I look at the different basic science research groups, also in different theoretical departments or at the Szent Ágottai Research Center where we are, this one is usually covered finding new targets, understanding new mechanisms. The second part, this one is also usually covered by the departments of pharmacology and also drug companies, and sometimes these different research groups has connections with the different drug companies and try to develop drugs. However, there is a real problem at the moment in the third part of translational medicine in the clinic, because unfortunately, currently, this is almost restricted to 95% understanding or analyzing retrospective data. If a students come to do a project, for example, to get the medical degree, going to a clinician, so usually the clinicians say, let's go to the Meds hall and write out retrospectively the data, which of course will be not a good quality and totally retrospective way. And unfortunately, this part, including meta-analysis, prospective data analysis, observational, interventional, clinical trial, usually not used if you look at the publications from Eastern and Central Europe. But of course, you can't do translational medicine or understanding clinical questions missing these part of science. Of course, there are points which are for everyone, and of course, there are ways which are only for clinicians. Okay, so question number 6. Today we start in translational medicine teaching the meta-analysis first. Why we start with meta-analysis? why we don't start with the cohort analysis or other. This is also very easy. There is no restrictions, so basic scientists, clinical scientists can work together. This is what we have done in the last 2 years with the Department of Pathophysiology and Gastroenterology. Really easy to learn. You already have the data somewhere, so your data collections is quicker than doing other part. This is really identify the holes in our knowledge and excellent learning method of the good publication practice. Here you read huge amount of publications and you immediately can realize that there are huge amount of data you cannot use because the bad publication process. So this is the best way for you to learn how to publish articles, and relatively you get quicker answers whether you continue the research on the topic or you don't continue the research because you already have the knowledge. So if I look at the timeline about the research results coming to your answers, finding new drug, this is the longest way. It's usually 15, 20 years, very important. All of the points are important because science cannot be done without any of them. But this you have to understand. If your aim is a drug development, you need to be relatively young to get answer at the end of the day. Of course, there are others like basic science or clinical trials, which of course you need to prepare. You need preliminary experiments. You need to follow from unknown to known, what I showed you plenty of times going around. Of course, it takes time. So really massive good projects, either as a randomized clinical trial or in the basic field, usually more than 5 years, 6, 7, 8 years. One of the reasons, and we also need to think about the whole PhD programs, which is restricted to 3 or 4 years, and most of the time mentors choose not the high projects to run because the people need to have immediate publications and they are not patient about waiting for the good results. This is a problem in science, and meta-analysis can solve this problem as well. Concerning understanding registry data, this is a very high quality of human data which we can collect. It's usually when you decide to establish a registry. After that, it's maybe 1 year when you establish it. You need around 2-3 years to get enough number of data, and after you do the analysis. So registry analysis, you get a quicker answer. However, meta-analysis, this is the quickest way. We had, for example, a scientist who did a really good meta-analysis within 3-4 months. So, but definitely within a year, you can answer, within half a year, you can answer to your questions. Of course, this is the way that you didn't make the experiments by yourself, so you collect the different data. How can a meta-analysis finish? First, this is inconclusive. There are big heterogeneity in the data, and you cannot have a final conclusion. But you understand where is the lack of data. So either you can decide that there is absolutely no basic knowledge in it. In that case, if you are, for example, in a basic scientist, you go back to the laboratory and perform more experiments. If you understand that, for example, you can't see the association between the things because it's not only you have available trial data, but you don't even have actually cohort data. What you can do, let's establish a registry, look at the analysis of the cohort data, and after go to clinical trials. And if you realize that everything is ready, you have cohort analysis, basic knowledge, but nobody made a randomized clinical trial, go there. However, in some cases, the meta-analysis already made a final conclusion, what you do. In that case, you don't need to do science. You go to clinical practice. For example, if my clinical question is, if the patient has 200 blood pressure, whether shall I decrease it to get less stroke, or shouldn't I decrease it? If you do a meta-analysis, all the articles will show that lower blood pressure will result with lower stroke incidence rate. Therefore, it's already decided you don't need, and ethically is also not possible to do a trial, which means you ask the questions with high pressure, shall you decrease it? In that case, you don't need to go further from meta-analysis. You go to do clinical practice and education. This is called A level of evidence. The evidence level, you can say A only if there are randomized trials showing the results and you have meta-analysis showing that there is no heterogeneity in the literature. So meta-analysis is very good to start if you are, for example, a PhD student, or you start working in a theoretical department of clinical department, because you understand whether your question is already answered or not. And if not answered, where are the holes in science that you should cover? And if you are clinicians, but the hole is in basic science, let's try to find the basic scientific team to answer first the question. Or if you are a basic scientist and your question is clinical, let's find a collaborative team. This is the other major advantage of translational medicine, bring the two fields together. So what can a basic scientist learn by performing a meta-analysis? I would like to show an example which has been done in the last 2 years. For example, look at acute pancreatitis and arterial pH. Whether lowering arterial pH, how it affects on the outcome of acute pancreatitis. So one of the a faculty member of the Department of Pathophysiology did a meta-analysis. And András Garami clearly showed in a meta-regression curve that if the pH dropping down in the artery, this will be associated with much more severe pancreatitis. So the arterial pH and the severity going together. Okay, so however, from the meta-analysis, you couldn't understand whether the low pH will make the pancreatitis more severe, or the more severe pancreatitis make the blood pH lower. So where is your question? How the blood pH affecting pancreatitis? Where you go back from meta-analysis? You need to go to the basic side, yes? Because if you would like to understand the question, you need to do basic science. So we continued the study in the basic field, and Demese taught in Szeged, did experiments when first she induced metabolic acidosis and after developed the pancreatitis and backwards, and it's immediately turned out that in metabolic acidosis, if you induce pancreatitis, you will have much severe pancreatitis, and backwards where. So there is a clear vicious cycle that if the severity is going to the worst side, pH dropping, but the dropping pH will further. So what does it suggest for us? It's very easy. What will happen if you see that the patients get more severe pancreatitis? You can organize a randomized clinical trial, or also in the middle, first preclinical trial with animals, that you do an intervention, you give bicarbonate to the patients, to the blood, and look at whether you can prevent to going to a severe pancreatitis. So from a meta-analysis, we went back to the laboratory, and after we will go further to clinical trial. So you're really speeding up the whole process doing the meta-analysis. And this can finally definitely go to patients' benefit. Next question, I'm a clinician. Shall I do meta-analysis? So this is good for you or not? The answer is again yes. For example, it was a meta-analysis with, as I mentioned at the beginning, and maybe it's we go. So Katalin Marta, who started as a PhD student in the laboratory, performed a meta-analysis about the early energy intake, and all meta-analysis showed that both in multi-organ failure, intervention, mortality, the early enteral feeding were beneficial. So from this point, also as a PhD student, she could start publishing articles, and also started the Gulyás study, which is the general utilization of energy back to the patients, which is a randomized clinical trial, which after 5 years will answer the question. So meta-analysis also solved the problem of a PhD student who has high aims and she doesn't want to finish the research within 3 years. So we definitely, from the energy side, from acute pancreatitis, you were a clinician, you already could use the meta-analysis for patients' benefit. Okay, the next question about us, like a Center for Translational Medicine, who we are. We just were established in 2 years, and how we can help. Either you are a basic scientist or either you are a clinician. So this is, we are really proud that in Central and Eastern Europe, this is the first Center for Translational Medicine. We have 3 main units. One of them is in the clinical science. The questions always start from the patient side. This is in the first department of medicine, and there is a separate department called Department of Translational Medicine, which was established to treat patients, and all the patients are treated in this unit are in some of the trials. The second is the basic science at the Institute for Translational Medicine, which is the former Department of Pathophysiology. We would like to enhance the basic science and also establish new research teams which will aim to study different diseases. And one of the most importantly part is about the core research group, the multidisciplinary research group, which is led by Andrea Santesi, and this contains many different multidisciplinary areas. Of course, you need a medical group who will teach the different ways, and if you have a question about whether how can I perform meta-analysis, cohort analysis will help. Of course, you need very high quality data and operative data management group and clinical trial group, because you need ethical administration, you need data quality team, you need clinical research administrators. Because why do we say this? Because most of in the science, in the medical practice, everyone is doing the other's job. So a PhD student is asked to do biostatistics, and we put the data to an Excel file. The medical doctor pushing the patients to the CT, and while the doctor's there, maybe the nurse giving the medicine to the patient. So all the jobs are done, but everyone is doing the other's jobs. So one of the main aim of this core facility that everyone should do what is their best. Because if a PhD student doing a biostatisticians with no knowledge, this will end to nowhere. So I felt I know something in biostatistics until we established the research center. After 2 years, I know that I have absolutely no knowledge in biostatistics. We have an IT group. Why we need an IT group? Because it's not possible if you have many centers to collect data in Excel and sending around. Currently, in acute pancreatitis, we have 110 hospitals from 28 countries submitting the data. We have 1.5 million data at that moment from patients suffering from acute pancreatitis. You can't use Excel for that. You need continuously developing IT. And of course, you need a patients club, you need a PR unit, and you need to work together. And this will actually speed up the whole process. Okay, so what we are doing currently, one of them is here we established the center, and first when we submitted our first grant application, we tried to establish the Center for Translational Medicine, especially the very beginning in Szeged and Debrecen, and also on Wednesday we will make the first translational center in a hospital, which will be in Székesfehérvár. We will help giving administrators, PhD doctors and others to involve patients from the hospitals, and of course we would like to enhance for more centers, because basic science is totally different from clinical science. Basic science depends on how creative you are and how much money you have. If you have one idea and you have 100, you need 100 mice. It's easy, you have 10 ideas, you need 1,000 mice. So to get from 100 mice to 1,000 mice, you need more money, 10 times more money. In clinic, around in Page, we treat around 250 patients suffering from acute pancreatitis. Even if we announce a free alcohol in Page, it wouldn't be more than 300 or 350, wouldn't help the incidence rate. So if you have 10 ideas, you need 10 times more patients, you cannot do that. You need the multi-center things, and also the hospitals, very important. In the Dutch pancreatitis study group, which is one of the best, the very well established, field, they have 8 universities and 25 hospitals in one IT groups. And they are just publishing in a New England JAMA Lancet without any restriction. Second, we do meta-analysis. Within the last 2 years, we started 83, and of course, plenty of times you cannot finish the projects, but 54 will be finished. Already we published 14 articles, and some of them from some of the resulted in clinical trials, or as you could see, basic science. 3 already accepted, and the other are in different phases. We also making different study groups, because you need a network, you need study groups. We already started with the pancreatitis study group, but now in endoscopy, this is also we have an ERCP registry, so we are very welcome if you have an idea, either basic scientist or clinical scientist, you have a research interest field. Now we have an IT system. Within 1 or 2 months, we can establish a registry, which will help you to provide huge amount of data for cohort analysis. This is the registry, so if you have a team, we will come back today. We are speaking about meta-analysis, but we have also an educational meeting how to establish registries or doing clinical trials. Currently, we have also many registries and trying building up. In the GINOP project, we will have 3 cardiology registries, cardiomyopathy, atrial fibrillation, and acute myocardial infarction. Concerning neuroscience, we will have a stroke registry, and in gastroenterology, we will cover almost all of the part. And of course, when you have the data, you need to put it to patients' benefit. You need to do clinical trials, randomized, observational, any kind of trials. So we are running now 5 different trials, and hopefully 3 or 4 other trials will start. So what is our main aim? First, to develop translational medicine in Eastern and Central Europe, apply the techniques we know, and share the knowledge of translational medicine for you, and offer the service of our core facility. So you need to come with your specific question, and we will try to help you to choose the methodology, which one you need to choose to answer the question. So the next question is, of course, question number 11. What is meta-analysis? We were speaking. However, I must say that there are much better people knowing about meta-analysis, who are biostatisticians or medical doctors performing that. So this is the course. We will be here. And Joel Sakac, who is a PhD student and doing very well the meta-analysis, he's the director of the course. And you will have today plenty of presentations from the clinical questions, how to raise it, and what methodology you need to choose, until the showing, like Aaron, very high quality meta-analysis for you. And finally, when we close the day, we will give you a time schedule that if you wish to join with an idea of doing a meta-analysis, how you can join this, and within half a year, we may, or hopefully we will, answer your research questions, either to go a basic science, go to applied science, or go to clinical science. So I would like to thank you for coming. It's a great pleasure to see a room like with full people, and I just would like to say that enjoy the course, and I would like to hand over the microphone to Joel. Thank you very much for your attention. But